Hey guys, it's me, Dr. Ree, and in this video we are going to be reviewing the central nervous system. Now, in lab, we went into this in-depth lecture lab about the central nervous system. In this video, I'm going to be doing a briefer version of this, so make sure that you refer to what the notes we took in lab for any other questions you have. So the central nervous system, just to kind of define what is it, is made up of the brain, and the spinal cord. And that is what this video is going to focus on. In our next video, or the next series of videos, which will be our next lab, we will look at the peripheral nervous system, which is the PNS. Now, we just divide the central the nervous system into the central nervous system, peripheral nervous system, but we can also divide it into 2 or 3 functional nervous systems, and those are the somatic nervous system, the autonomic nervous system, and the enteric nervous system. The somatic nervous system is all about sensation coming in, going to the brain, and then motor information or motor action coming out. The autonomic nervous system is your fight or flight and rest and digest systems, or your sympathetics and parasympathetics. We will talk all about that in another lab. And the enteric nervous system is the digestive system. We can also talk about nervous tissue by if it's gray matter or white matter. White matter is made up of mostly myelinated axons, whereas gray matter is mostly made up of unmyelinated axons and cell bodies. This will make more sense when we get into what is an axon, what is a cell body, and so forth. So let's zoom in and look at the different cells of the central nervous system. There are 2 categories of cells. We have neurons, which are the main functional unit. So this is our functional unit of nervous tissue. And we have glial cells, which are the sidekicks or the helpers. We're going to talk about the glial cells in a second, but let's focus on the neuron first. So here's my little drawing of the neuron. We call this part right here the soma, which is the fancy way of saying cell body. And we call this part right here the axon. Now, in lab, I explained how I think of a neuron like an airport. And I'm not going to go into that long explanation now, but let's just label some of those parts that we talked about from the airport. So when I get to the airport and I come in via my dendrites. So all of these are dendrites, and those are where information comes in. Then we have right here, that is our cell body or our soma, and then they go to security, and we called security the axon hillock. This is where we get our decision of whether or not a action potential or a message is going to go to the next cell or is going to be sent through the axon. Then we travel down our long road, which is our axon, and we can do that faster if we use those floor escalators. And we call those, which kind of look like marshmallows here, our myelin sheaths. Myelin sheaths are created by glial cells, and we'll talk about what glial cells create these myelin sheaths in a second. So how these myelin sheaths work, if I kind of zoomed in on this, is so each of the boxes or the rectangles is a myelin sheath, and this space in between the 2 is known as a node of Ranvier. And when the ions, specifically sodium ions, come in, you'll talk more about that in class. When those ions come in, they can't get out when they're in the myelin. And so they freak out and they go, okay, I need to get out of here. I need to get out of here. So they run as fast as they can, Speedy Gonzales style, to the next node so they can go out. Then they come in and then they freak out. They go as fast as they can to the next node and so forth. The reason why we have to have nodes is that if they came in to a hall that was just dark forever and they never could see the light, they'll give up and they'll stop moving. So that's why we have those nodes, to give them a little light to guide them so that they want to go there, so they quickly go there. They can go up to 100 times faster through the myelin than they do on an unmyelinated axon. Once they take that long road to the end, they reach the axon terminal. And that's what we have right here. And the axon terminal is where we will have a synapse. And that is where the message will be sent on to the next receptor, whether that is another neuron or it is a receptor in the body. And we can see some of those parts in this image right here. So you see all these glial that are pointed out here. Let's talk about or define 6 different types of glial cells now. So these are our sidekicks. Our first one is an astrocyte. An astrocyte is I didn't spell that right, astrocyte. This is the most abundant glial cell in the central nervous system. It does like everything. It does general support, nutrient support, and it creates the blood-brain barrier. So astrocytes do a lot. Our next are ependymal cells. Ependymal cells, right, kind of look like epithelium. And that is because they are also linings. They line the ventricles, which means they are in, I added an N there, ignore that N, which means they are in the central nervous system, and their job is to produce and maintain cerebrospinal fluid, which is abbreviated CSF, cerebrospinal fluid. Our next is microglial cells. Microglia are our immune system. So these are our macrophages of the central nervous system. So its location is the central nervous system. Now down here we have our 2 myelinators. This one is Mr. Ollie. This is my oligodendrocyte. And over here is my Schwann cell. My oligodendrocyte is what creates myelin in the central nervous system. And my Schwann cell creates myelin in my peripheral nervous system. Now you can see in this image that an oligodendrocyte produces multiple myelin sheaths, whereas my Schwann cell is a 1-to-1 ratio. It's 1 Schwann cell per 1 myelin. Schwann is a 1 myelin man. And our last cell is a satellite cell. In a satellite cell, I probably spelled that wrong. Again, spelling doesn't count because I can't spell. Satellite cells are going to be very similar to astrocytes in the central nervous system, but they are general support in the peripheral nervous system. So in these images, you can see these glial cells and neurons together. So again, here we have, here it is. So here we have our astrocyte, we have our axon terminal right here. So that's kind of what we're seeing. So astrocyte, we have our oligodendrocyte, our microglial, and our ependymal. And in the peripheral nervous system, we have our Schwann cells and our satellite cells. Now let's go from our zoomed in to our zoom out, and let's talk about our brain. So again, I'm going to be going way quicker through this material than I did in lab. So let's just do the general organization of the brain. So we have our cerebrum on top. that is divided into two hemispheres. Right? We have one hemisphere here and one hemisphere here. Each of those hemispheres is then broken down into lobes, and we're going to talk about the 4 lobes that we break it down into. Now, we can't see it in any of these pictures, but our next kind of station after our cerebrum is the cerebrum goes to, I can't draw it on here, so I'm just going to, so our cerebrum goes to our diencephalon, which is made up of our thalamus, our hypothalamus, and our epithalamus. Then it's going to go to our brainstem, and our brainstem is made up of 3 components. The most superior one is our midbrain. The next is our pons, and the lowest or the most inferior is our medulla oblongata, which I swear is not a Star Wars character. And so we can see here's our midbrain, here's our pons, and here's our medulla oblongata. And then from that, it reaches our spinal cord. Now there are other structures like the cerebellum, like the limbic system, things like that, like basal ganglia, that are kind of within those, but those are our general structures, specifically when we think about the somatic nervous system. So, starting in the cerebrum, we have 4 lobes, so each hemisphere is divided into 4 lobes. So let's look at each of them. Our first lobe is right here in the front, and we call that our frontal lobe. In our frontal lobe, we have our prefrontal cortex, so that's where we get a lot of decision making. That is where there's centers that are responsible for addiction, things like that. We also have on our dominant side an area called our Broca's area, which is responsible for motor of speech. And right here at the back, we have our precentral gyrus, which is where we will find our primary motor cortex. So when I think frontal lobe, I think behavior and motor. Behind our frontal lobe is our, I think I did it in blue in the lab, is our parietal lobe. And it starts at right behind our precentral gyrus at our postcentral gyrus. And it kind of exudes back here until it reaches the occipital area. So some of the other regions that we have in here that we talked about was Wernicke's area, which is about right here. And Wernicke's is responsible for comprehension of language. And then we also, like I said here, have our postcentral gyrus, which is where we have our primary, which is where we have our primary somatosensory cortex, which is our cortex that tells me I'm feeling things. When I understand that I am holding something and this pen is smooth, that kind of idea. So parietal lobe is about sensation. It also is an area where we have logic and comprehension of things like math. This is like analytical, our analytical lobe. Underneath our frontal and parietal, we have our temporal lobe. Right here. There is a superior temporal, an inferior temporal, and a middle temporal gyrus that we see here. The temporal lobe is responsible for, or is in our primary auditory. Auditory means hearing. our primary olfaction. Olfaction is our smell. And this is also where we have memory. So hearing, smell, and memory. What a great. Also music, right? Hearing, music, all of that is kind of in this area. And then our occipital lobe is in the back, right where we had our occipital bone. And that cortex is all about vision. We as humans dedicate an entire lobe of our brain to vision. It's where our primary visual cortex is, occipital lobe. So those are our 4 lobes of the brain. Again, our cerebrum is divided into 2 hemispheres, and each hemisphere has a frontal lobe. Each hemisphere has a parietal lobe. Each hemisphere has a temporal lobe. Each hemisphere has an occipital lobe. Now, I talked about the ventricles before, and our ventricles are what are holding our cerebral spinal fluid. So this is our fluid for our structures. This is where our nutrients are coming from for the different structures of our brain. And so with that, this is the complicated drawing of it. I'm going to do a little uncomplicated my way of remembering what ventricles relate to what regions. So starting with our cerebrum that we just talked about, we just said like multiple times that it has 2 hemispheres. So I like to draw my 2 little kind of hemisphere sides, and those are known as my lateral ventricles. We have a left and a right one. And those are related to my cerebrum. Then we come down into our next region, which is our 3rd ventricle, and that is related to our diencephalon. The diencephalon is made up of our thalamus, hypothalamus, and epithalamus, and the subthalamus, but don't worry about that. That 3rd ventricle is then going to flow into our cerebral aqueduct. Which supplies our midbrain. Our midbrain is the most superior part of our brainstem. And then that flows into my little baseball diamond, which is my 4th ventricle, and that is what's going to supply our cerebellum as well as our pons and our medulla oblongata. So here we can kind of see, so our ventricles, like if we put this together, our lateral would be on the side here, and then we would reach into our 3rd ventricle. Here we're looking at our diencephalon, so like I said, so our thalamus right here, that is a smaller 2 lobes, it has 2 large lobes that acts as the relay station to the somatosensory cortex, which is right here in that postcentral gyrus. And so that is where it's kind of the relay station for information coming from the spinal cord before it goes to the cerebrum. We also have our hypothalamus. This is going to do all of our regulation kind of things. So hunger, thirst, water balance, hormone balance, sex drive, all of that's coming from our hypothalamus. And then back here is our epithalamus, and that is made up of our pineal glands and our choroid plexus. Our pineal gland is all about circadian rhythms. This is where melatonin as well as some hormones involved in sexual maturation are produced. So that's the diencephalon. So cerebrum, then diencephalon, and then underneath that, so right here, here, and here, this whole thing right here is our brainstem. So you can see there's our brainstem right there. Behind the brainstem is our cerebellum. So here's our occipital lobe. It's right underneath. Our cerebellum is all about motor learning, balance, equilibrium. It's an amazing part of our brain, but we're not going to talk too much about it. I'll leave that for lecture, but that is where the cerebellum is. And let's just define the parts of the brainstem. So we have our midbrain, our pons, and our medulla oblongata, which you can see here in this image, and then the diencephalon on top. And those are going to run into the spinal cord, which is where we're going to end today, is talking about the spinal cord. So the spinal cord is going to start after our medulla oblongata, and it's going to go down until around L1, L2 of the lumbar spine, right? And it's going to travel through the vertebral foramen that we saw in our vertebrae. So when I draw some of the structures that are related to the spinal cord, I start by drawing, I have this cervical enlargement. Then it continues down through that vertebral foramen, through the thoracic spine until it reaches near the lumbar spine, and then it has this lumbar enlargement. And then at the very end, near L1, L2, so right after the lumbar enlargement, we have this kind of cone-shaped ending, and we call this cone-shaped ending our conus medullaris. But we still need nerves to get down to the legs and such, right? So what happens is at the end, at conus medullaris, we have a whole bunch of nerves that come out of it. So I kind of draw them like a tail, and that is because its name is horsetail, or the quadra equina. So the collection of nerves after the conus medullaris at the end of the spinal cord is known as the quadra equina. Now the spinal cord, if it's not tethered down, if it's not held down, it could recoil up like a slinky into our brain, and we do not want that. So we have this thing called filum terminale, and it's this extension of the meninges, that kind of links it down or holds it down or tethers it down, and we call that the filum terminale. Now, I just used the word meninges, and it's like, okay, what are the meninges? Well, we learned that there are 3 types of meninges. Our meninges have layers, just like onions. So our thickest, most outer layer is our dura mater. Then we have our next layer. It's not as thick, it's more porous, or it gets its name because it kind of looks like spider webs. And that next layer, our middle layer, is our arachnoid layer. And then our super thin layer, our thinnest layer, and our most inner layer is our pia mater. And the pia mater is attached to the spinal cord via these little tiny teeth-like filaments called denticulate filaments. Denticulate filaments. Now, if I drew a quick spinal cord like that, there's that quadra equina, and I took a little nice cut of it transversely or horizontally, you could use either term, right in the middle. Let's draw what a cross section of the spinal cord looks like. So if I was looking at a cross section of the spinal cord, I don't want that pen, I want this pen. It sorta kinda looks like this. And then on the inside, we kind of have this SpongeBob flower kind of look. Whoops, my pen moved. We have this ventral white commissure in the middle, and then we have some nerve stuff coming out. I'm going to define these parts in a second, but I'm just going to draw them. Okay. So this would be on both sides, so I could draw this on this opposite side too. Okay. Now let's define some of these parts. So right here, this is our horn, so we're going to say that this is our posterior aspect, and this is our anterior. Remember that the other terms we used for posterior and anterior, our back, is our dorsal, and anterior, our front, is also our ventral. I'm going to use the terms ventral and dorsal because in neuroscience, those are the terms that are seen most often. So our dorsal or posterior horn is right here. So this is our horn. This one would be our dorsal horn. This one would be our ventral horn. Right here. Those horns are attached when they come out into our roots. So this one, because it's on the dorsal side, is called our dorsal root. And this one on our ventral side is called our ventral root. our roots come together into a spinal nerve. And then that spinal nerve separates into our rami. The dorsal rami are going to go to the back of the body to supply the muscles and things in the back. And the ventral rami is going to go to the front. As well as the ventral rami typically will also supply a lot of our limbs and things like that. So those are the anatomic. Now, when we talked about the somatic nervous system, we said that sensory information was coming in and motor was coming out. And so we're going to kind of go through the motions that we talked about of how that somatic nervous system works so that you understand the properties or the what is found in each of these locations, because it's very applicable clinically or professionally. So here is my hand, and say in my hand I decided to touch a flame. Ow, that hurts, okay? So we're starting with ow, that hurts, I touched something. So that sensation that I'm like, oh, wow, ow, that hurts, that information is going to be first received by receptors in our hand. And those receptors are then going to go up through my arm, up through into my rami, from my rami into my spinal nerve. Now sensory in the back. Sensory in the back, that's what I want you to think of. I sense things in the back. So it's going to come up, it's going to go into my dorsal root, and it's going to send that information to the cells, cell bodies, the somas, in my dorsal horn. Now, sensory neurons, and you'll learn this in lecture, are bipolar neurons. So these sensory neurons that are in, or peripheral neurons that are in my PNS that sense things, those type of neurons are called bipolar, and that's because they have two axons. So we have an axon here and an axon here. And the soma is in the middle of the two. We call the area where those soma for those sensory neurons the dorsal root ganglion. Okay, so like I said, so that's sending that information. It sends it to the dorsal horn. There's cell bodies here, and those cell bodies send their axons up to the brain. So once it tells that, oh man, that hurt, it's going to send it up to the brain. It's going to reach the thalamus. It's going to cross over to the other side, and then it's going to go to the parietal lobe, right, to that post-central gyrus where we have our primary somatosensory cortex, and that's where it's going to have the sensation of, oh, ow, that hurts, okay? From there, I decide, what should I do? Oh, I should move my hand out of the way, okay? This is more of a reflex thing. I shouldn't have done fire. But let's say it's a basketball and I feel the basketball, okay? And what's my reaction? I'm going to hit the basketball down so that I dribble it. So that motor coming out is going to come down from the brain, cross over to the other side, and it's going to send its information to the somas or the cell bodies in the ventral horn. Those cell bodies are connected to our peripheral nervous system, or a part of our peripheral nervous system, come out of our ventral root, through our spinal nerve, back through the rami, to my hand, and I dribble the ball. So motor is always going to be in the front, or you can think of it's ventral. And sensory is in the back, or it's dorsal. And that's even in the brain, right? The parietal lobe is in the back, and that one had sensation, and the frontal lobe is in the front, and it had motor. So now, using those examples that are in your workbook, if we go through them, right, the dorsal horn and the ventral horn both have cell bodies. The dorsal horn has cell bodies for sensation, and the ventral horn has cell bodies for motor. Our roots both have axons. The dorsal has sensation axons. The ventral root has motor axons. The dorsal root ganglion has cell bodies for sensation, and then our spinal nerve has both sensation and motor for, or in the version of axons. So these are where those axons for both of them go through. So that was my super quick version of what we did in lecture. If you have any questions, please let me know, and I will see you in lab.